I'm going to try and put genetic genealogy in the context of what I understand Fiona has been talking to you about in the first term. I'm going to give you the standard caveats as to why you might not want to be involved, but they don't bother me and I hope they won't bother you. Go through the definitions of the basic components of DNA, how they're inherited, mutations and recombination. I'd love to show you all the DNA websites, but there'll probably be only time to look at one or two. And this is all leading up to my favorite case study from the cases I've worked on, how the grandchildren of a man who was known only to his grandchildren as Eugene Lynch from Ireland used their DNA to identify him 110 years after he disappeared from California. So I'll refer ahead once or twice to the example that's coming at the end. And I'll try and watch the clock so that I have time to go through the example. Your DNA is just another source for genealogy. People say nowadays that not using your DNA for your genealogical research is like deciding you're not going to use census returns. It's that critical. I hope to tell you how to get as much evidence and information as possible from your DNA results and how to combine the DNA results with your other sources. You're the expert on your own family history. You have the oral traditions that you learned growing up in your memory. Hopefully you have some family papers in your attic or wherever they're kept, and you have your family DNA in every cell of your body. And your task as a genealogist is to reconcile those three categories of private information with each other and with the public information that Fiona's been teaching you about. And sometimes you'll find they agree, and sometimes you'll find there's a disagreement. One of the things that we talk about in genetic genealogy is the NPE, not the parent expected. And there's other ways of laying it out. And you may get a surprise for your DNA results and find that at some point in the family tree, the DNA doesn't fit with the written record. So you've got to be prepared for those surprises if you're going to send your DNA off for analysis. We're the first generation that has had digital records created of our DNA. The more people are in the DNA databases, there's now tens of millions, the more valuable those databases are going to be for other people. Just to give you an idea of, hopefully my links are going to work, quite how big these databases are. The biggest company is Ancestry DNA, which is now over 20 million customers. Next biggest is 23andMe, which is about 12 million. MyHeritage about 5 million, GEDmatch and Family Tree DNA 1 to 2 million, Living DNA I think is about 300,000 people. It's affiliated with Find My Past, and I believe you all have Find My Past access for this course. The more people we can get in these databases, the more we'll be able to reconstruct our ancestors' DNA from our own DNA. Your descendants will be eternally grateful to you for leaving them your DNA. You will be cursing your ancestors for not leaving you their DNA. There are companies now trying to extract DNA from artifacts. So if you have any family letters, hairbrushes, toothbrushes, anything like that, guard them carefully and stop them from getting contaminated with living people's DNA, and eventually it may become commercially viable to get your ancestors' DNA extracted. There's one case of one of my own DNA matches who was a foundling, and her father had been narrowed down to one of two brothers. Somebody produced a letter that had been sent by one of the brothers, and living DNA managed to extract the sender's DNA. The DNA of whoever licked the envelope or licked the stamp. So we presume that was the man who wrote the letter and confirmed which brother was her father. So it can be done in special cases, but it's not really commercially viable yet unless you're very wealthy. And the companies who are promising to do it are still struggling with the technology, I think. So your DNA, and if you're lucky to still have parents or grandparents alive, you have only half of your parents' autosomal DNA and a quarter of your grandparents. So anybody from an earlier generation, their DNA is even more valuable to you in your research. When you're combining your DNA with your family tree, I strongly recommend that you put what you know about your ancestry in a genealogy database. Ancestral Quest is the one that I use. That's what my pedigree chart looks like in Ancestral Quest. The people who see that they are a DNA match to you will want to know who is the common ancestor from whom they have inherited the DNA. If they can see a pedigree chart like that with 5 or 6 generations of your ancestors, it could become obvious immediately who the common ancestor is. Make sure you have at least 1 generation of your deceased ancestors on the pedigree chart linked to your DNA, and then at least your matches can work back themselves and see who the common ancestor might be. But if you have no pedigree chart linked to your DNA, or if everybody in the pedigree chart is marked private or living, that's not much good to your matches. That's Ancestral Quest. There's lots of other similar programs like Roots Magic, Reunion, Family Tree Builder. Reunion is great on the phone. If I can't bring the laptop, I have the whole database on the iPhone and I can look it up there. Wikipedia has a page with dozens of software packages listed. I used to be a great fan of building your own family tree in your own software on your own computer, until mine got so big as to be almost unmanageable because I kept spreading out and adding cousins and cousins of cousins and in-laws and DNA matches that I couldn't quite link up to. And broadband has got much better, so online sites are now a feasible alternative. I like, in particular, wikitree.com because it has DNA features. There's 3 single large world trees which try to have a single profile for everyone who has ever lived. Wikitree, Family Search, which is affiliated with the Mormon Church, and Genie.com. Wikitree is the only one that has built-in DNA facilities. So before I even come to explaining the components of DNA, there's a picture from Wikitree showing from whom I've inherited each component. It lists the ancestors from whom I got my Y chromosome, the ancestors from whom I got my mitochondrial DNA, the ancestors from whom I got my X chromosome, and all my ancestors can contribute to my autosomal DNA. I'll come back to those different types of DNA in due course. Lots of people like to put their family trees on Ancestry.com. Ancestry.com has a great business model. It lures in new customers. It says, fill in your family tree. If you don't know, just guess. And then it takes those guesses and it sells them on to the next wave of customers as hints. So unfortunately, Ancestry might be fine as long as you ignore the hints, which are other people's wrong guesses, of which there are many. And Ancestry and MyHeritage are also trying to combine all those user-donated family trees into their own internal single-world tree in an effort to explain DNA matches. Hasn't worked very well so far. Sometimes it's great, sometimes it gives you nonsense hints. If Ancestry gives you a nonsense hint, there's nothing you can do about it. It can still be there 10 years later. If MyHeritage gives you a nonsense hint, you can mark it as rejected or nonsense, but it still stays there marked as rejected. So beware of those big commercial sites that are not always as good at getting accurate family trees as other sites like Wikitree, which has an emphasis on accuracy and sources. I have another page, that's probably the link to the page that tells you why and how to get the most out of your DNA results. It's invariably going to be out of date because all the websites change things around every now and again, but it will give you a guideline as to how to fish in all the gene pools. Put your DNA on all the DNA websites. I call what I discovered helping a lady called Mrs. Murphy, who was adopted. I call it Murphy's Law of Genetic Genealogy. There's half a dozen big DNA websites. And I say if there are n DNA websites and your DNA is in n-1 of them, your most critical match will be in the nth one. So this lady was on all the websites except 23andMe, and her full sibling was on 23andMe, and it took her a long time to find him, because that was the only one he was on. Caveats. I've already mentioned foundlings and other such things. DNA has let the genie out of the bottle as regards family secrets, adoptions, fostering, things like that. If there is a secret like that in your family, as you'll see in the case study, keep your DNA out of the DNA databases if you want it to stay a secret. The world has changed. There's no reason why these things should stay secret, but they're still very emotional. As you can see, 110 years on from a man disappearing in California, DNA found who he was and where he came from. Didn't find where he went to yet, but we're still hoping that somebody else will show up. And remember, there's social workers and all sorts of support services for people who do find surprising results. Treat family secrets as sensitively and sympathetically as possible. Other people have reservations because they're afraid insurance companies will get their hands on your DNA. There's this great thing in America called the Genetic Information Non-Discrimination Act, GINA, which says that a DNA company may not discriminate against somebody, an insurance company may not discriminate against somebody with bad DNA by charging them higher premiums. Converse is the DNA must discriminate against people with good DNA by charging them the same as people with bad DNA instead of charging them lower premiums. But if you think about it, if you have, crudely speaking, if you have bad DNA and you're likely to be very ill and die very young, your health insurance will be a lot more expensive, but your pension will be a lot cheaper. Whereas if you have good DNA and are going to live forever and never have to go to hospital, the insurance company that knows that will give you cheap health insurance, but they'll charge you a lot more for your pension. So what you lose on the swings, you gain on the roundabouts. So it doesn't bother me what insurance companies know about me. The other reservation people have is the use of DNA by the law enforcement authorities. Investigative genetic genealogy, which has been used mostly in North America to solve cold cases, serious criminal cases, murders, rapes, and things like that, from DNA collected at a crime scene. So that DNA can be put into the genealogy databases, just as an adoptee will put his or her DNA into the genealogy databases looking for close relatives. And if they find close relatives of the suspect in the crime, or maybe just unidentified remains found somewhere, they can work out, is there anyone in this family that was in the right place at the right time to have gone missing and not been found, or to have committed the crime. Most of us probably don't have any problem with criminals being brought before the law, but some of us might have problems with the death penalty, or with maybe infanticide being considered as a hanging offence or something. So people don't like, some people don't like their DNA getting into the hands of law enforcement. You can opt out of that with some of the companies. But anyway, it's just something to bear in mind. And the final caveat, people think that they can put their DNA into a genealogy database and remain anonymous and get some assistance. There can be absolutely no assistance to you if you don't tell your DNA matches who you are. At least tell them your birth surname. And as I said, put up a family tree that goes back to at least one generation of deceased ancestors, so they can work from them on their own from there if necessary. DNA and communities. When you send your DNA into one of these companies, they will turn your spit or your cheek swab into a computer file. And they'll run that computer file against all their other customers, and they will sort the other customers by how closely they are related to you. So you will get a match list. I'll show you a match list later on. And then it's your task to figure out how you're related to the people on those match lists. What they also do is they peddle estimated ethnicity percentages. We all know, I hope, at this stage that if you go back 100 or 200 years, most marriages happen between people living within 5 or 10 miles of each other. So if you take a small area, especially an island or a peninsula on the west coast or something, everybody's going to be related to everybody else within a few generations if they're not moving around very much. So you will find that your DNA matches are from the same area as your ancestors, even if you can't go back far enough with the records that have survived to work out exactly how you're related. In other words, people whose ancestors lived close to each other geographically now end up close to each other genetically. As a converse of that, we hear about the Irish and the Basques, for example, sharing DNA. So if you find common DNA between people living in Ireland and people living in the Basque country, yes, they're related, but that tells you absolutely nothing about the direction of migration. It could be that both populations migrated from completely different place to where they ended up, or the migration could have gone in any direction. Genetics doesn't tell you about migration. You have to have other historical evidence to deduce how the DNA came to be similar in two far apart locations. The ethnicity percentages that are sold by the DNA companies, they're their main marketing tool. They're very popular in countries like the United States, where the vast majority of the population is descended from immigrants who've arrived within the last couple of hundred years. They're basically based on reference populations of people with what Sean Ennis in Genomics Medicine Ireland came up with this lovely tag. He calls them GAA people, people with genetically anchored or geographically anchored ancestry, people who can prove all 8 great-grandparents or all 16 great-great-grandparents all lived within a small radius. They're going to be related to everybody else whose ancestors lived in that radius. So these estimates are very sensitive to the reference population that is used. They come with huge margins of error. I liken them to an opinion poll. When you read about an opinion poll before an election, you're told these numbers have a margin of error of plus or minus 3%. There's nobody regulating the DNA industry to say you have to tell your customers what the margin of error around these percentages are. I did work with 1 or 2 cases where the ethnicity percentages were helpful. I had a match the other day and I found her obituary and it said she was born. Well, I found the mother's obituary, the match's mother's obituary, and it said the mother was born in Japan. And I said, well, I'm obviously related to the father. And then I looked in another of the DNA websites and I found, no, I am related to the mother who was born in Japan. But I was able to look at her estimated ethnicity percentages and I said 29% Irish. So I said, obviously, some Irish-American soldier in Japan after World War II got up to some sort of mischief or whatever, because she was using a Japanese surname, but her father had a very Irish surname. Well, there was another one that I dealt with many years ago, an adoptee living in Dublin, adopted in Ireland, assumed she was going to get 100% Irish ethnicity. She got 33% Italian. And when we did a little bit of research, we discovered her birth mother was big into classical music and opera and so on. And her birth father was an Italian opera singer who was a guest star with the Dublin Grand Opera Society. And we found his name advertised in the newspaper archives 9 months before this lady was born. But unfortunately, he had a wife and family back in Italy, so his Irish girlfriend was left in the lurch. So that's the selling point. That's what's got 20 million Americans into these databases, but it's not of any huge help to us with our genealogy, which is what's a big help to us are the lists of matches we get. The other types of communities that we have in the DNA world are the DNA projects run by FamilyTreeDNA.com, one of the big DNA companies based in Texas. I'm an administrator or a co-administrator of all these projects. One is called the Clare Roots Project. A lot of my ancestry is from County Clare. So it's got 1,863 members, all with some ancestry in County Clare. It has an activity feed like a chat room where people can chat. You can look at the DNA results of other members of the project. And I try and give a little bit of advice there. So that's the sort of project you might join if you want to correspond with people whose ancestors are from the same area. The other type of project, second type of project is surname-based. The Y chromosome descends from father to son in the same way as in our Western society, the surname typically descends from father to son. So people with the same surname will have a similar Y chromosome, but you will actually find, here's the Clancy project, which I'm a co-administrator of. I'll just bring up the DNA results page and look at the colorized chart. You will actually find for common surnames that there can be multiple different genetic origins. For all sorts of reasons, we've had a surname era for about 1,000 years now in Ireland. There's many opportunities for surname DNA switches, where for whatever reason, somewhere in those 30 generations since surnames were introduced, a son has taken maybe his mother's surname, maybe his stepfather's surname. As somebody said to me recently, maybe the mother has been cuckolded, maybe there was a cuckoo in the nest, maybe somebody running away from the law or running away from a family, like our friend Eugene that we'll come to later, had decided to adopt a different surname. There's many reasons why the surname won't follow from one generation to the next, even if there's only a 1% or 2% chance in each generation of a switch. Over 30 generations, that adds up. That gives you many different genetic origins for a single surname today. Or if you take something like an occupational surname like Smith or Miller, lots of unrelated Smiths and Millers might have taken their occupation as a surname. Even in the case of Clancy, in the old annals going back to the 1300s, there were two Clancy families, one in Doolin in northwest Clare, one in Chadderee near Newmarket and Fergus in southeast Clare. And the medieval historians tended to assume, well, this must all be the one family. They're in what's now the same county. They might be 50 miles apart. But we discovered, I've color-coded these. So basically the people in yellow are from one genetic origin, the people in the orangey colour from another, the people in the blue colour from another. There were Clancys also in Leitrim. So the Leitrim Clancys, the Southeast Clare Clancys, and the Northwest Clare Clancys have three different genetic origins. The surname was adopted independently in those three areas. And DNA has enabled us to confirm that, which even some medieval historians were surprised by. Then the third type are haplogroup based. A haplogroup is just a group of people with similar DNA. So the Y chromosome changes a little, but not very much over the centuries. So you will find that you have, if you're a male with a Y chromosome, you have Y DNA matches where the common ancestor might be 1500 or 2000 years ago and didn't use any surname, because surnames only came in roughly at the time of Brian Boru, who died in 1014 at the Battle of Clontarf. The O'Briens claim O'Brien is the oldest surname in the world, and it derives from the Irish O'Brien, grandson of Brian, based on Brian Boru. And in fact, the present Lord Inchiquin, Conor O'Brien, 32 generations descendant of Brian Boru, has been an early adopter of DNA analysis. So there's a very active O'Brien DNA project and an associated L226 project. The Irish annals tell us that there are other surnames, mostly around County Clare, where Brian Boru originated, that derive from a common ancestor with the O'Briens. They're called the Dalgash. The common ancestor is Cos, who lived maybe 1500, 2000 years ago. And it has been found that the surnames that, according to the annals, descend from Cos do have similar DNA. And this is the L226 mutation, has been attributed more or less to Cos or to some male around his time. And there's a group of people with that DNA who discuss what are the related and unrelated surnames. And those surname projects are like those run by the Guilds of One-Name Studies, or like the clans involved in Clans of Ireland. So I've talked a little bit about the different components of the DNA and what they might be used for. So I'm going to come back now and talk in a little more detail about exactly what our DNA is made up of and which parts can be used for which purposes. I said I'd better give you a definition of genetic genealogy. The Wikipedia one is the best one I could come up with. The use of genealogical DNA tests, i.e. DNA profiling and DNA testing in combination with traditional genealogical methods to infer genetic relationships between individuals. I'm a statistician. To me, testing is testing a statistical hypothesis. What the DNA companies do with your DNA is extracting data from it. It's not testing hypotheses. Here's our friend Eugene again in WOTO. WOTO is short for what are the odds? And this is a chart which allows you to insert hypotheses on a family tree. So here's our friend Eugene, and here's his grandfather, and we're trying to figure out which of the grandfather's children is Eugene's father. So we slot Eugene in under each child. We slot in all the DNA matches that we have found, the beige colored cells. We slot in the hypotheses as to where Eugene's grandchild might fit. And the numbers tell us, well, this hypothesis is in red. It's impossible. It's not consistent with the DNA evidence. This one is not consistent with the DNA evidence. The other 4 in green are consistent with the DNA evidence. Some are more likely than others. The least likely gets a score of 1. The one with the extra green around it down at the bottom gets a score of 255. Its hypothesis 6 is 255 times more likely than hypothesis 1, based on the DNA from one of Eugene's grandchildren. But we have 8 of Eugene's grandchildren to look at. They will all give us different rankings of the hypotheses. And as we see at the end, between all 8, we were able to rule out all the hypotheses except 1. So when you look at your DNA matches, you come up with a theory. This might be how I'm related to this person. Then you bung all the numbers into the Watto tool, and it tells you just how much more likely your theory is than other competing theories, or which of your competing theories is more likely. DNA stands for deoxyribonucleic acid. You might need that for a pub quiz. You don't need it for genealogy. It's made up of nucleotides, adenine, cytosine, guanine, and thymine. Again, that's for the pub quiz. They get turned into letters on the computer, A, C, G, and T. And you get back a computer file like this. Each gene, each location in the DNA gets an identifier like RS, a string of digits, of no genealogical significance. The DNA is broken up into 23 pairs of chromosomes. So the second column is the chromosome number. Each of those pairs of chromosomes has many millions of locations on it. The third column is the location along the chromosome. The last column is the results. In this case, we're looking at an autosomal chromosome, which has a pair of letters, 1 letter from the father, 1 letter from the mother. So at this location, the person we're looking at got an A from the father, an A from the mother. Down here, the person got an A from one parent and a G from the other parent. Unfortunately, the technology can't tell which parent the A came from and which parent the G came from. And that means that we're dealing with statistical inference rather than hard conclusions a lot of the time. Where do these letters come from? We all start off as a sperm fertilizing an egg. The sperm from our father, if we're male, brings a Y chromosome. If we're female, it brings an X chromosome. So the male side determines whether the child is male or female. But the father, through the sperm, also provides 22 autosomal chromosomes. I'll show you a picture of what the chromosome breakdown looks like later. The mother then in the egg provides everyone with an X chromosome. So if you're a male, you have a Y chromosome and an X, one from your Y from your father, X from your mother. If you have a female, you have 2 X's, one from each parent. The egg provides another 22 autosomal chromosomes, so the paternal 22 and the maternal 22 pair up together. And the egg from the mother also provides what's called the mitochondria. So each of these components has a different inheritance path. Well, let me go back and just look at my pedigree chart, for example. I'm a male, I have 1 Y chromosome, I'm a Waldron, it came from my father who was a Waldron, came from his father who was a Waldron, came from his father who was a Waldron, came from his father who was a Waldron. I have mitochondrial DNA, it came from my mother, who was a Durkin. It came from her mother, who by coincidence was a Durkin before and after marriage. It came from her mother, who was an O'Neill. It came from her mother, who was a Mrs. O'Neill, whose maiden name I still don't know. But my 22 pairs of autosomal chromosomes came 1 of each pair from my father, 1 from my mother. So exactly half from each parent. The half from my father came partly from his father, partly from his mother. And on average, a quarter of the 22 pairs of autosomal chromosomes came from each of my grandparents. On average, 1/8 came from each of my 8 great-grandparents. on average 1/16 came from each of my great-great-grandparents, and 1/32 and 1/64. And eventually the autosomal DNA dilutes down so much that you have ancestors who are your genealogical ancestors, but they're no longer your genetic ancestors because you inherit a negligible or zero amount of their autosomal DNA if you go back 10, 20, 30 generations. Whereas the Y chromosome comes down unchanged in its entirety from the direct male line or direct patrilineal ancestor, the mitochondrial DNA comes down unchanged and intact in the direct female or matrilineal line, on which the surname changes every generation. So that's the hardest part of your genealogy typically to research. Just briefly to show you the X chromosome. Where's my X chromosome link? It's slightly messier. If I'm a man, I have one X chromosome. I'm blue. My female ancestors are pink, male ancestors are blue. So my X chromosome, the only one I have, came from my mother because my father provided my X to pair with it. My mother had two X chromosomes, one from each of my maternal grandparents, one there in blue, one there in pink. The previous generation, my maternal grandfather had one X from his mother. My maternal grandmother, well, she's female, had two X's. So basically the colored cells are the only ones from which the X DNA can inherit. That's the advanced class. We'll talk more about X DNA next year. Mutations. Wait for time. Not too bad. So far, I have said that DNA is inherited by transcription from parent to child. There will occasionally be a small little transcription error. At one location, one letter out of many millions might change. There are some locations that change very frequently. They're the ones that the police have historically looked at in their criminal databases, because if the location changes very frequently and they look at dozens or hundreds of these locations, it's very unlikely that more than one person will have exactly the same pattern. So for identifying a criminal beyond a reasonable doubt, you want to look at the fast mutating locations, but they change too fast to be of much genealogical use. For genealogy, we want those that mutate maybe once in many generations, or once in the history of mankind is even better. And they're good for genealogy and for identifying people who are closely related. But the vast majority of the billions of locations on the chromosomes are identical for all humans and for apes and chimpanzees and orangutans and other primates as well. So most of our DNA is identical, but the tiny fraction that varies from one person to another is still enough to be of great use to us in genealogy. There's two types of mutations that we particularly look at. One is called a short tandem repeat. It means a short string of letters, maybe 4 letters like CCTG, which repeats itself several times, typically on the Y chromosome that passes from father to son. And you might find that at one generation, the son has an extra repeat, so might have 8 CCTGs instead of 7. And then that will be passed on to his male line descendants from there on. And that was the first marker on the Y chromosome that was used for genetic genealogy about 20 years ago. The other 3-letter acronym you'll see is the SNP. That just means a single letter has changed between the father and the son, or between the parent and the child, if it's on one of the other chromosomes. So we'll go back. I showed you the Clancy project earlier. This is the Marinin project, another surname project I'm involved in. Marinin is another County Clare surname, much less common. And in fact, we've found, with 2 exceptions, I think, all the Marinins in the project have very similar Y chromosomes. There are 111 columns here stretching off the screen to my right. You're right as well over that side. And in this layout, within each subgroup, there are colors to highlight where there's a difference between the men and the subgroup. So as you can see, in each of the Marinin groups, there's very few colored cells. But down at the bottom, we have this group with the beige header, men with female Marinin ancestors. No reason for them to have the same Y chromosome. They have different surnames. You have lots of colors there. to indicate differences in the STR mutations. That was the early way of doing this. The SNP mutations, which are still more expensive to analyze, have become much more powerful in recent years. And that haplogroup column tells you the most recent confirmed SNP mutation that each group of men has. So we have combined in the subgroups men with exactly the same most recent SNP mutation. And the strange thing is there's very few cases here where we have two people with a single common ancestor. Actually, in that group we do. But if you know your Clare geography, that group, they're all from Curracodee, so that mutation basically ties that branch of the Marinans down to one townland. This group are all from Shrath townland. That mutation ties that branch of the Marinans to Shrath townland between Kilkee and Kilrush and Doonbeg. And it's almost magic that you can look at a mutation and say, I know what parish your ancestor came from. So a lot of these are men whose ancestors went to America 150 years ago, had no idea where in Ireland, let alone where in Clare they came from. But the Y-DNA SNP analysis can tell them what parish or what townland their ancestor came from. We don't have the primary sources to say exactly how the Marinans who lived in Curracodee 200 years ago were related to each other, but they have the same DNA and they lived in the same townland. So to tell how closely related people are based on the Y chromosome, how closely related two men are, we have the concept of genetic distance. Basically, we look in that group at how many colored cells there are, how many differences in the 111 or the 67 or the 37 markers that they've had analyzed there are. And that number of differences, one or two little quirks, is called the genetic distance between them. So two men with a genetic distance of 9 out of 111 are going to have a common ancestor well within the surname era, maybe within the last 200 or 300 years. Way back in the early days, 0 out of 12 was considered significant, but the common ancestor, looking at only 12 markers with no differences, could be 2,000 years ago. So the smaller the distance, or the smaller the number of differences between the two men, the closer the relationship. Let me look briefly at the tree of mankind. The theory of this is that at some point, a couple of 100,000 years ago, there was only one man alive who has direct male line descendants still alive today. There could have been many other men alive at the same time whose male line has died out or daughtered out because some man along the way had no sons and only had a daughter. So that man has not been identified, but he's referred to as the Y Adam. A bit like Adam in the Bible, except that Adam in the Bible was the only man in the world, not the only one with living male line descendants today. And eventually one of his descendants had a mutation which split the tree into haplogroup A and haplogroup B, and then that split with another mutation. And eventually, 20 years ago, the people doing this said, oh, there's about 20 different types of Y chromosomes in the world, and that's all we need. That tree, which didn't even need all the letters of the alphabet, has now grown to a tree with 50,000 branches on it. So that's the speed at which Y chromosome analysis has grown in the last 20 years. And that's why those haplogroup numbers you saw all have 5 and 6 digits in them, because there are hundreds of thousands, well, 50,000 branches, and within those branches there might be 3 or 4 mutations that are always found together, never found separately. There is a link there to the full haploid tree. It's large. It'll take the rest of the lecture just to download, so I'm not going to show it to you. In the case of autosomal DNA, it gets shuffled around or recombined from generation to generation. There is a little stylized example that I put together. Every cell in my body, for example, might have that blue string of letters that I got from my father, and on the corresponding autosomal chromosome, a string of letters in red that I got from my mother. Every sperm and every egg is potentially unique, because while the cells in the body have pairs of chromosomes, the egg and the sperm have just 22 single chromosomes rather than 22 pairs. And they're formed by crossovers or recombination between the paternal and the maternal in each pair. So one particular sperm or one particular egg might have a string of blue letters that comes from the father, then a string of red that comes from the mother, then a string of blue that comes from the father, then a string of red that comes from the mother. And those crossover points are random and unique. There's one every 19 letters, I think, in that little example. It's actually about one every 19,000 letters is the actual frequency of the crossovers. So I'd run halfway up to Kingsbridge Station if I were to put all the letters together. So that's why we all have such different autosomal chromosomes. And to look at how closely two people are related when we compare the autosomal chromosomes, we measure how much of those 22 pairs of chromosomes they have in common. The unit used for that is called the centimorgan. It's one of the mysteries of genetic genealogy is where the centimorgan scale comes from. It's never questioned except by me. I think a lot of the quirks are down to the centimorgan scale maybe being wrong. It runs from 0 up to about 3,600. So there's various charts showing you how to turn these centimorgan numbers into relationships. This one shows 3,600 is a parent-child relationship or an identical twin relationship. Anything between 2,400 and 3,200 is full siblings. Between about 2,400 and 2,000, we don't know whether we're looking at full siblings or half siblings. We could be looking at full siblings who share a little less than average or half siblings who share a little more than average. So at 2,250, it's equally likely you're looking at full siblings and half siblings. Then there's another range where you're definitely looking at the half sibling category, which also includes similar relationships like grandparent-grandchild. And then there's a range down here where you're definitely looking at first cousin relationship. You need a little more than the DNA to distinguish between first cousin and great uncle, great nephew, for example. They're going to share the same amount of DNA, but two first cousins are going to be about the same age. Great uncle and great nephew are going to be 60 years apart in age. And when we get down to the bottom, 50 or 60 centimorgans, the relationship can be extremely distant. But if you share over 100, you're going to be third or fourth cousin or closer. And certainly for a DNA match with over 100 shared, if you're a good genealogist, you should be able to work out the relationship. There's various other ways of visualizing it. There is a chart showing the amounts shared by different relationships, parent-child in the middle with 3,500, sibling with 2,500, half-sibling with 1,800, and so on. And the nicest one, I'm just going to put in 209.5, because that's the number we're going to meet in a minute when I come to the example. Except this one has decided not to open for me. Come on, load up. There we go. So there's the same chart we looked at, but now there's a box at the top, and you can type in a number, and it calculates 209 shared is either in the second cousin category, or the second cousin once removed category, or possibly just third cousin, or possibly even closer, but roughly equally likely to be from the second cousin or second cousin once removed category. And then if you scroll down, it shows the chart with the possible relationships highlighted and the impossible relationships grayed out. I've done a comparison table of the 6 big DNA websites. I encourage you to fish in all the gene pools. You can send your spit to Ancestry.com. You can send your spit to 23andMe. You can download a data file derived from your spit from both of those websites, and you can upload it free for the basic services to GEDmatch, to Family Tree DNA, to MyHeritage, and to Living DNA. You can pay a little extra on most of those sites if you want access to additional facilities. I've put in the customer numbers across the bottom. Today's prices, but they vary from time to time. MyHeritage has a sale on now. Valentine's Day is coming up in a couple of weeks. There'll be another sale for that. Black Friday, Cyber Monday, whatever is the best time of year to buy these, especially the Y-DNA analysis that costs a lot more than the autosomal analysis. Let me just show you GEDmatch for a couple of reasons. I finally suddenly realised there's 21 people watching me online, far more than here in person. And I prefer the classic GEDmatch, which I've been using for years. I hate when people change websites around and don't improve them. There's some differences. There's some things are better, some things are worse. You have a menu when you log in of different things you can look at. The one-to-many comparison result is the one that I like to use. Everybody gets a kit number. You can put in your own kit number, and it will bring up your 3,000 closest matches. These are my matches. The top 5 are all my first cousins. Then the 6th one, Deirdre, is just a little below the first cousins, but she's only a second cousin. She's actually a double second cousin because my grandfather and his brother married 2 sisters. And she's a little more than a double second cousin because my grandfather was an identical twin. So it's as if, genetically, it's as if we have the same grandfather and our grandmothers were sisters. Anthea Ring down here, you can Google her name to discover the sad story of how she was found abandoned as a baby 80 odd years ago, and eventually using DNA identified her parents. That's the case I talked about, finding the DNA on the seal of an envelope. So my comparison to her, just to give you a visual impression of what chromosomes look like. It's all color-coded. A full match means related on both sides, father and mother related. That will be green. A half match will be yellow, one related through one parent only. A red means no relationship at all. And shades of blue mean definitely a close relationship. So on the first chromosome, it's a mixture of red, no relation, and yellow. We might be related, but the yellow isn't long enough to say there's definitely a relationship. Small little bits of yellow are not significant. Longer bits of yellow are significant. On the second chromosome, there's a long bit of yellow with a couple of little bits of green. Little bits of green don't tell us anything. Just by coincidence, there's a bit of DNA shared by my father with Anthea, as well as a long bit shared by my mother with Anthea. But the blue underneath there says that's definitely a match. We definitely shared 23 centimorgans in chromosome 2, which came from some common ancestor. We made this great breakthrough working out Anthea's parents. Gone back to her great-grandparents. The records run out. I still have no idea exactly how I'm related to her. Probably third cousins. We share nothing much on the other chromosomes. Chromosomes are not all the same length as you might think from that picture. It's just scaled to take advantage of the width of the screen. How are we for time? I'll just briefly show you Ancestry. Those of you who haven't seen a match list, these are my matches from Ancestry. Lots of ways we can analyze them. There's a new thing recently where it is trying to tell you which parent each match is related through. There's an older thing called Through Lines, which goes back and uses the user-donated family trees to guess who the common ancestor is. A lot of those hints will be wrong. You can search by all sorts of fields, including the family trees. You can add stars and colored dots to tell you what side of the family they came from. And again, closest relation at the top. And there's a total of how many? If I go by shared DNA, it says I have 18,879 matches. I'm not going to go through all those. So for the last 5 minutes, let me go back and talk about the case study. You can read all the other stuff at your leisure. It's up there on my website. I'll put up the URL again, and I'll put it in. Actually, why don't I do it now? Because otherwise I'd forget. For those of you watching on Zoom, if I copy that and go to the chat and paste it in, you should be able to click on that link and read these notes yourself at your leisure. So if you're in the room, write it down. If you're watching on Zoom, click on it and bookmark it when the Zoom call is over. So let me jump down to the case study. There we go. This is how I combined oral knowledge, archival records, and DNA to identify this mysterious Eugene Lynch. He came from the townland of Moveen. You will all have seen it in history books, in the Illustrated London News during the famine era. go. Moving. Might have seen that sketch. That's the townland we're talking about. He lived somewhere up there on the hill, or his ancestors lived somewhere up there on the hill. I knew that there were Lynches living in this house in Griffith's valuation. The local legend is that farm, which I've marked Mary Lynch in the Griffiths map, was divided between two sisters. Their maiden name was O'Dea. One married Lynch, one married O'Connell. Eventually the O'Connells were evicted and the Lynches took over the whole farm. Mary's son is in Griffith's valuation down there. We know she must have been a widow in Griffith's valuation because the farm would have been in her husband's name if he was still alive. Her son down here was known as Porrig, Mora, Nance. Porrig for Patrick, Mora, Mary was his mother's name. Nance must have been her mother's name, must have been Mrs. O'Dea that we hear about in the local folklore. His brother was Tomás Mora Nance and inherited the mother's farm eventually. We had to go to America to a death certificate in Connecticut to find out that the parents of Tomás and Porrig were Michael Lynch and Mary O'Dea, or O'Dea as we would call it in Ireland. Through the Clare Roots project, I was asked to recruit descendants of the Lynches to give their DNA, and one of them duly did. Meanwhile, this family in California told us the story of grandfather Eugene Lynch. Bad grandpa, they call him. I call him Eugene the Blackguard. He appeared out of nowhere. The story was he abandoned the pregnant grandmother before the fourth child was born, but he still put his name down in the census in 1910, a few days before the child was born. So there's a slight mystery as to exactly when he disappeared, but he abandoned the family. Apparently there's no marriage record that gives his parents' names, but there's no birth record because they didn't know where he came from, and there's no death record because he disappeared. So we're left with DNA to try and identify him. Here is a DNA match between two people, I'll just call them Claire and Ed. Claire was a lady I swabbed in West Clare. They share 50 centimorgans on the 8th chromosome. We know Claire is Mrs. Lynch's descendant. Ed is Mrs. O'Connell's descendant. The DNA that they share must have come from one of their common great-great-great-grandparents who were Mr. and Mrs. O'Dea. We don't know Mr. O'Dea's first name. Mrs. O'Dea must have been Nance because that was the matronymic that her grandchildren had. Michael Lynch in California, Eugene's grandson, shares 27 centimorgans of that. So he must be descended from or related to Mr. or Mrs. O'Dea. But his surname is Lynch, and one of their daughters married Lynch. So he must be descended from Michael Lynch and Mary O'Dea. His grandfather Eugene is too young to be a child of Michael Lynch and Mary O'Dea, which shares too much DNA with their other descendants to be anything more distant than a grandchild. So we put together this autosomal matrix showing the DNA shared by 8 grandchildren of Eugene in California and many descendants of other Lynches. And we have some huge numbers here in the 200s for people who are only third cousins once removed. I'm being told I have to finish up. So basically these people were on the next plane to Ireland, came to see where their ancestor lived. We couldn't figure out who was that missing generation between Eugene and Michael Lynch and Mary O'Dea who had to be his grandparents. We went around swabbing the rest of the descendants, and there was none of those children standing out, none of the sons standing out as Eugene's father. But eventually I had this brainwave. What if Eugene was using his mother's surname? What if Michael Lynch and Mary O'Dea were Eugene Lynch's maternal grandparents? I went looking in the workhouse records to see was there a single mother called Lynch with a son called Eugene. No joy. Then I looked, but we do know of one sister. She was Mrs. Curry. She had a son called Eugene. He was born about the same time as Eugene Lynch. He went to America about the same time as Eugene Lynch. He settled down, he married a girl from home, and they had 2 children in New York. He went west to find his fortune and said to the wife and children, I will send for you later. And they never heard from him again for 110 years until I reunited the 2 families. So that's the power of DNA.